For over a century, humans have been trying to perfect the art of splitting water into hydrogen and oxygen. We've built complex machines from electrolyzers lined with expensive platinum catalysts to high-pressure systems powered by wind and solar energy. But nature has been doing this quietly for billions of years, using microscopic enzymes that can split water, move electrons, and make hydrogen with astonishing efficiency. They're called hydrogenases, and they could hold the key to unlocking the next revolution in green hydrogen production. Most people think hydrogen technology begins with electricity and ends with a shiny industrial plant. In truth, it starts with biology. Deep inside certain bacteria and algae, there are molecular machines so efficient that they can produce hydrogen from water without heat, pressure, or precious metals. These enzymes, microscopic and invisible, achieve in a cell what humans can barely do with an entire factory. And that's what makes them fascinating. Hydrogenases are nature's own electrolysis systems, operating on scales smaller than a grain of dust, yet performing chemistry that rivals some of our most advanced energy technologies. The first hydrogenase enzyme was discovered in the 1930s when scientists noticed that some microorganisms released tiny amounts of hydrogen gas while metabolizing. At first, they thought it was a fluke. Later, they realized that these microbes were carrying out a reaction at the heart of hydrogen energy itself, splitting protons and electrons to make molecular hydrogen, H2. Since then, researchers have identified hundreds of different hydrogenases across bacteria, archaea, and algae. Some of them can generate hydrogen directly from water, while others recycle it as part of their own metabolism. Either way, they reveal a natural blueprint for low-cost, low-energy hydrogen production. What makes hydrogenases remarkable is their chemistry. Most industrial electrolyzers rely on rare metals like platinum or iridium, because those are some of the few materials that can efficiently split water into hydrogen and oxygen without corroding. But hydrogenases use earth-abundant metals, mainly iron and nickel, arranged in intricate three-dimensional structures inside the protein. These metal centers act as catalytic hearts, channeling electrons with near-perfect precision. They can produce hydrogen at rates comparable to engineered catalysts, but at room temperature and without expensive materials. In fact, some hydrogenases can operate at turnover frequencies exceeding 10,000 reactions per second, a level of activity that puts many artificial systems to shame. In simple terms, these enzymes do what our machines do, but faster, cleaner, and smarter. They achieve this through bio-inspired design, balancing electrons and protons in perfect synchrony. Imagine a microscopic assembly line where each component, from metal atoms to surrounding amino acids, is optimized by evolution to carry out hydrogen conversion with minimal energy loss. There's no heat, no pressure, no large-scale equipment, and yet the reaction just flows. The question scientists are asking now is, can we learn from these natural systems to improve our industrial ones? Can we create hybrid electrolyzers that use bio-inspired catalysts instead of costly rare metals? This idea, sometimes called biocatalytic hydrogen production, is at the cutting edge of hydrogen research. It blends chemistry, biology, and materials science into one goal: to mimic what nature already perfected long ago. The challenge, however, lies in stability. Most hydrogenases are fragile. They evolved in specific environmental niches: oxygen-free conditions, moderate temperatures, and precise pH levels. Expose them to oxygen or heat, and they denature, losing their catalytic activity. Industrial electrolyzers, on the other hand, operate in harsher environments with fluctuating voltages and gas pressures. So, before we can integrate hydrogenases into technology, we need to make them tougher. That's where synthetic biology comes in. Scientists are re-engineering these enzymes, modifying their amino acid sequences, or embedding them into protective materials like carbon nanotubes and metal organic frameworks. The goal is to combine nature's precision with human durability. One promising direction is the development of bio-hybrid electrodes. These systems use biological catalysts attached to conductive surfaces, allowing enzymes to perform their natural function while electricity drives the reaction. In laboratory tests, such electrodes have achieved impressive current densities and efficiency levels, proving that bio-inspired designs can rival traditional electrocatalysts. Some teams have even connected hydrogenase-based systems directly to solar cells, creating sunlight-driven biophotocatalytic devices that split water with remarkable efficiency. Another exciting line of research focuses on hydrogenase mimicry. Instead of using the whole enzyme, scientists study its active site, the tiny molecular cluster where hydrogen is formed, and replicate it synthetically. These model catalysts often use iron and sulfur in the same geometric arrangements found in the natural enzyme. They're simpler, more stable, and easier to mass produce. By bridging biology and inorganic chemistry, researchers hope to design catalysts that work as well as nature's versions but survive in industrial settings. There's also growing interest in using microbial electrolysis cells, where living bacteria themselves act as miniature electrolyzers. In these systems, microbes take in electricity and produce hydrogen directly, powered by renewable energy. It's a form of living electrolysis, and while still experimental, it suggests that future hydrogen plants might not only be mechanical but biological, with engineered microbes continuously generating hydrogen as part of their metabolism. Why does this matter? Because the current generation of electrolyzers, alkaline, PEM, and solid oxide, still face major cost and material challenges. Iridium, for instance, is one of the rarest elements on Earth, and global production is measured in mere tons per year. If hydrogen production scales up to terawatt levels, iridium supply simply can't keep pace. Nature's catalysts, on the other hand, use elements found almost everywhere: iron, nickel, and sulfur. If we could replace even part of today's precious metal catalyst with bio-inspired alternatives, the cost curve for hydrogen could drop dramatically. Beyond cost, hydrogenases also offer insights into energy efficiency. Every electron in their system is accounted for. There's almost no wasted energy, no heat loss, and no side reactions. In industrial terms, that translates to lower overpotentials, less voltage required to produce the same amount of hydrogen. It's the difference between brute force engineering and molecular elegance. When you compare these natural systems to man-made ones, you start to see how biology can teach us new design principles for sustainability. One of the most studied enzymes, called Phe hydrogenase, contains a delicate iron-sulfur cluster at its core. This cluster acts as a molecular switch, accepting electrons and protons and recombining them into hydrogen molecules. Another type, knife hydrogenase, is more robust and can even operate in slightly oxygenated conditions. Understanding these mechanisms at the atomic level requires advanced tools like X-ray crystallography and ultra-fast spectroscopy. Each discovery reveals not just how hydrogenases work, but how evolution solved energy conversion long before we invented electricity. In recent years, researchers have started combining the best of both worlds, using bio-inspired electrocatalysts and conventional electrolyzers. These hybrid systems have shown surprisingly strong performance. Some experimental electrodes coated with hydrogenase fragments or mimics have achieved efficiencies close to platinum at a fraction of the cost. While still early in development, these breakthroughs suggest that biology and engineering can work hand in hand to push hydrogen technology forward. The potential applications go beyond energy production. In the field of carbon recycling, for instance, hydrogenases can help drive reactions that combine hydrogen and CO2 into synthetic fuels. Because these enzymes can shuttle protons and electrons so efficiently, they're ideal for coupling with carbon-fixing enzymes like format dehydrogenase. Together, they form miniature biological factories that transform waste carbon into fuel. A process that, if scaled, could make carbon-neutral chemistry a reality. Still, the road from lab discovery to industrial adoption is long. Enzymes don't last forever, they degrade over time. Large-scale production would require bioreactors capable of maintaining enzyme stability and activity under variable conditions. Yet, the lessons learned from hydrogenases are already influencing material design. Many modern catalysts now mimic enzyme architecture, building in proton channels, molecular scaffolds, and electron relays that resemble the natural systems. Even if pure enzyme technology never replaces metal catalysts entirely, its influence is reshaping how we think about efficiency and sustainability in hydrogen production. There's also a deeper philosophical point here. We often assume that innovation means inventing something entirely new, but sometimes the most profound innovations come from observing what already exists in nature. Hydrogenases remind us that the planet has been running clean energy cycles for eons. Photosynthesis, respiration, and microbial hydrogen metabolism are all forms of natural energy management. In studying these systems, we're not just copying them. We're learning to participate in the same cycles more intelligently. For example, certain green algae use hydrogenases during photosynthesis to balance excess electrons, releasing small bursts of hydrogen under specific conditions. Scientists are experimenting with genetic modifications that could increase this output, turning algae into microscopic hydrogen factories. Others are exploring artificial photosynthesis systems that integrate hydrogenase-like catalysts with light-absorbing materials. The idea is to create solar panels that not only generate electricity but also produce storable hydrogen fuel directly from sunlight to fuel with nature's efficiency built in. The intersection of biology and clean energy opens up a new way of thinking about technology. Instead of massive centralized infrastructure, imagine distributed biohydrogen units. Self-sustaining modular systems powered by engineered microbes or enzyme reactors. They could operate on waste streams, agricultural runoff, or even sunlight, quietly generating hydrogen without complex machinery. This isn't science fiction. Prototypes already exist in research labs from Europe to Japan. The challenge now is scaling them economically and ensuring long-term stability. If we look at the broader hydrogen ecosystem, from renewable electricity to fuel cells, industrial use, and synthetic fuels, biological catalysis could play a supporting but crucial role. It won't replace electrolyzers overnight, but it could supplement them where cost, temperature, or material constraints make traditional systems less viable. Over time, hybrid approaches, combining enzymes, advanced materials, and AI-driven control systems, might give us the best of all worlds: the speed of machines, the intelligence of biology, and the adaptability of software. In many ways, the story of hydrogenases mirrors the evolution of clean technology itself. Early discoveries seemed obscure, isolated in labs and papers, but as global attention turns to sustainable energy, what once looked like curiosity-driven biology now appears deeply relevant. The enzymes that quietly power microbes might one day help power cities. The implications go beyond energy. Learning from biological hydrogen systems could also reduce our dependence on rare materials, create new green industries, and even inspire educational programs that merge biotechnology with renewable engineering. It's a convergence of disciplines that reflects the complexity of the world we're trying to decarbonize. As we push toward net zero, we often focus on massive infrastructure: gigafactories, pipelines, and electrolyzer farms. But maybe the next big leap will come from something microscopic. An enzyme so small it fits inside a single cell, yet so powerful it could redefine how we make hydrogen. Nature's own hydrogen factories show us that it's possible to achieve high performance without high cost, using design principles honed over billions of years. So the next time you hear about hydrogen megaprojects, remember that innovation doesn't only happen at industrial scale. Sometimes it happens in the quiet chemistry of life itself, where electrons and protons dance under molecular precision, and where evolution has already built the perfect machine for splitting water. The future of hydrogen may be written not just in blueprints and budgets, but in enzymes and DNA. By studying these natural catalysts, we're uncovering what could become the next generation of clean energy technology, one that blurs the line between biology and engineering, between living systems and machines. And that's a story worth watching closely because it reminds us that the solutions to our biggest energy challenges may already exist inside the smallest forms of life. To explore how breakthroughs like these connect to real-world hydrogen project development, from feasibility modeling to finance and design, visit H2Hub, ReneEnergy.com. Learn how biology, technology, and finance are converging to build the next generation of sustainable hydrogen projects worldwide.